Microsoft PowerPoint to translate the English into Spanish. So if there's mistakes, email Microsoft, it's their problem. Okay, so I'm going to talk about the impact, potential impact of climate change on soil biology and biochemical properties of soils. So some of it's hypothetical because there's not many experiments, so I'm going to talk about the potential impact on the soil biology and give some examples. So the outline, I'm going to talk about some of the projections of climate change, including temperature, precipitation, but more importantly, extreme events. I think that's critical for the impact of climate on soils. Then I'm going to talk a little bit about the impact on soil biology and give an example of that, but then use some experiments that we're working on to look at the effect of land use and precipitation gradients on the soil biology and soil activity. So if we look at climate change, let's see, can I do this? Okay. This shows the temperature record for the last basically 2,000 years. And what you see here is that temperature has oscillated through those 2,000 years up until about the last 100 or 150 years. And in that last 150 years, there's been a dramatic increase in the temperature record. And we can correlate that to carbon dioxide as well. So we've seen, not only is it higher than it's been in the last 2,000 years, but the rate of increase is extremely rapid, which causes dramatic impacts on vegetation, on life, and of course then on soils. So the projections from the IPCC are shown here for temperature and precipitation. And if you look on the right-hand side of the panel, this is the projections for the end of the century under a high emission scenario, which is the pathway that we're on currently. And so what you see here is basically the globe is warming, although not evenly. There are parts, particularly the high latitudes, that are going to see up to 9 to 11 degrees centigrade increase in those regions, so the polar regions particularly. See here in Colombia, what's that, probably about a 4 to 5 degree potentially increase. But on the lower panel is the precipitation. And I think that's critical, at least from a soils perspective, precipitation change is going to be more dramatic on soils and soil biology than the temperature increase. And what you see here is that, well, the storyline is that the wet areas are going to get wetter and the dry areas are going to get drier. And if we look where agricultural production occurs here in U.S., Central America, parts of South America, Australia, and Europe, they're going to see less precipitation. And Colombia is, right about there. They're kind of on a dividing line between that. But it's not just precipitation. It's the seasonality. And sorry, I don't have a slide for South America or for Colombia, but what you see here is change in soil moisture by season. And at least for the North American continent, what we're seeing is that the summers are going to get drier across the whole North American continent. Here, winters, drier winters, but in the northern part, wetter. Again, showing that weather projection for that. But basically, because of the higher temperatures, it's going to get drier. but it changes by season. The other factor is extreme events. And what you see here is the change in precipitation intensity. Again, this is from the IPCC, and there's going to be an increase in the intensity of that precipitation events. For North America or for Canada, that may mean more snowstorms. Even though it's warmer, it's still going to freeze, and so you may get more intense snowstorm events. But for the world as a whole, generally, we're going to see more intense rainstorm. And in fact, but even with the precipitation increase in those intense storm events, because of the warmer temperatures, we're going to see an increase in the number of dry days. And that's pretty much true for the mid-latitudes. Again, Columbia is kind of on that dividing line. May see the same or slightly drier number of dry days. But if you look at the U.S., and again, I'm sorry about the U.S. example, but this is precipitation events here that exceed a 2-day precipitation event, so those intense storm events. And what you see here for the last century is that we have seen an increase in the number of those extreme events, those heavy precipitation events. We're also seeing an increase in the number of record high days, again showing that temperature and moisture relationship. What does that mean? Well, a pictorial picture view of that is illustrated here. 4 years ago, in Kansas, we saw record droughts and record heat. And so we couldn't even grow maize for 2 or 3 years in a row. And of course, then we had bare soil. That resulted in this event here in Kansas. This is not from the 1930s. This is from 2012, 2013, because of these dry events. But last year, on September 3rd in Kansas, we had 20 centimeters of rainfall in 4 hours. 20 centimeters of rainfall in 4 hours. That same year, in 2018, we were in drought. We made up all rainfall in 4 hours. So we had drought and we have floods in the same year. Again, those extreme events. And this just shows an example of those heavy rainstorm events in Oxisol in Brazil. So our soils are going to be vulnerable. Our urban areas are going to get flooding, and our soils are going to erode away under those extreme events. So those are some of the projections, and actually what's already happening. And so we don't have many experiments where we can test the effect of climate change on our soils. But we do have a few experiments out there, and this is one experiment from Kansas where we manipulated the rainfall. So we wanted to mimic what those intense rainstorm events, but dry periods in between those rainstorm events. So we had the same amount of rainfall, but we capture that rainfall, let's see, in these rainout shelters, capture that rainfall, and then held it and put it on in fewer events, but more extreme events, more intense precipitations. So we stored and reapplied onto, this is grassland, and we wanted to look at the extent that rainfall patterns, these intense events, would have on ecology. So we were allowed to take some soil samples from these plots. And so what you see here is just an example where we looked at the effect on this rainfall intensity. This is normal rainfall, and this is the more intense rainstorm events, precipitation events. And interestingly, what we saw was that we saw a change in the microbiome, the microbiology. we saw an increase in fungal abundance. But because of that change, we saw an increase in carbon use efficiency. Fungi are more efficient with carbon than bacteria. So actually, we didn't see a change in the short time period, 8 to 10 years. We didn't see a change in carbon, but we did see a change in the microbial community and more efficient use of that carbon. The effect on other nutrients we weren't able to study, but we would anticipate that nutrient cycling, nitrogen, phosphorus would potentially be altered with that change in the microbial community. So that's just an example of potential climate change effects on the microbiome. So the other example that we have is using our natural precipitation gradient. So Kansas is located here. Across Kansas, we have this natural precipitation gradient. We go from over 1,000 millimeters in rainfall in the east to a little over 400 millimeters in the west. And so we have this new project that we're looking at the effect of the precipitation gradient and land use on the microbiome of aquatics, plants, and soils. So there's a group looking at the effect on water microbiome, aquatic, the effect on the plants, and then on the soils. Across that gradient, Kansas, we have the opportunity to compare land use. So much of Kansas, about 10 million hectares, is in prairie. And in Manhattan, this is the tallgrass prairie that was grazed by bison 150 years ago. Nowadays, it's mostly cattle. The thing about the prairie is it has a very deep root system, extends down to several meters into the soil. Much of Kansas, another 10 million hectares, is in cropland. Mostly wheat, but also maize, soya, and sorghum. The thing that's different is the rooting depth of those annual crops is much less. Extends only to less than a meter in many cases, where the prairie extend over 2-3 meters. So differences in rooting depth with that land use and also precipitation. So what we did that we're currently doing, this is real on-time data, is that we've studying 4 locations across that state. I have data from 3 of those locations presented today. So we're looking at that gradient from 1,000 millimeters down to 483. I'm going to show you some data from Hayes, 580 millimeters, 850, and about 1,000 millimeters. And then we have 3 land uses. We have native prairie, cropland, and then cropland that's been converted back to prairie. Okay, so this is soil carbon at the top 5 centimeters at those 3 locations. And so we have the native prairie. There is a slight decrease in carbon as you get wetter. That's not what we would expect. It might be a change in vegetation as well. But if you look at the agriculture systems, what you can see is we've lost 60-70% of that carbon as a result of cultivation. But as you go wetter, the carbon in the soil increases, and that's what we normally would expect. But again, it's 50% or more less carbon in those agriculture sites. In the restored or replanted grasslands, we've recovered some of that carbon. So it's partway in between, between the native and the cropland. We've also looked at the microbial community, and this is just an example. We've got more extensive data. But what we see from phospholipid analysis is the soil microbial biomass, and one example is the mycorrhizal fungi. The rest of the fungi and other bacteria, other organisms show a similar response. But what we see here is in the native prairie, we got more microbial biomass as you get wetter. So that's in contrast to the carbon story. But in the agriculture site, here again, we have less than 50% of the microbial biomass in that agriculture site, of those sites. There's a little bit of effect of moisture, but not consistent. And the mycorrhizal fungi are showing the same response. An increase in mycorrhizae, arbuscular mycorrhizae, across that precipitation gradient, but we greatly lose that community in the ag systems, in the agriculture systems. The other thing is that we're interested in the relationship between the microbial community and the chemical and the physical properties of those soils. So in our lab, we're looking at aggregate stability, so the wet sieving technique. And what you see here is the prairie is dominated by macro aggregates greater than 2,000 microns in size. And that's typical of the prairie across the United States. Again, these are mollisols, and so approximately 60% of the soil is in these large macro aggregates. In the ag system, we've destroyed those aggregates. They're gone. Again, across that precipitation gradient, that tillage, intensive tillage breaks up those macro aggregates, and no effect of moisture on that. What we do see is that there's a strong relationship between the amount of mycorrhizal fungi and total fungi in the soil and aggregation, except for the agriculture sites. Agriculture has destroyed that relationship. Again, because we've lost the mycorrhizal fungi and lower fungi. In the tillage, we lose those large aggregates. And that's really critical because, as we talk about, that soil structure is important for those extreme events. When we have that heavy precipitation, those soils are more prone to erosion. They have lost that aggregation. Whoop. I didn't do that. There, okay. Okay, so the soil profile, so that's just at the surface. So what we're interested in is, what about the whole profile? So we've looked at going down through the profile down to 2 meters. So not 1 meter, 2 meters. And we're thankful to the USDA Natural Resource Conservation Service, NRCS, if you're familiar with that in the United States. They dug these pits, not me. They dug those pits, and we sampled those profiles down here. And this is a picture of this mollisol. I believe this is the one in Kanza in Manhattan, Kansas. So this is carbon through that profile of those 3 land uses. This is for the dry site, 580 millimeters, and the wetter site, 850 millimeters. So what you see here is the native prairie is in the red, and then the yellow, or I guess that's yellow, is the ag site. And what you again see is that we've lost carbon not only at the surface, but we've lost it throughout the soil profile, both for the dry and the wet sites. The other thing to note for those interested in soil pedogenesis, soil development, is that the agriculture site has undergone additional soil development. We got different horizons with the ag site. So agriculture has changed the pedology of that profile and created more horizons in that. We're still understanding that. But with the wetter site, we've lost carbon, but we haven't lost it as much as the dry site. And I can show you the nitrogen content, the same relationship, but what you see here is that the nitrogen has not been lost as much as the carbon, particularly down at depth. Here's on the wet site, this is the prairie. The nitrogen on the wet site, the agriculture is lower, as you expect, but what you actually see down deep is more nitrogen. And so the hypothesis is that result of tillage, nitrogen has been mineralized and then mobilized and moved down into those deeper horizons. And so we are accumulating nitrogen at depth. And in fact, if you look at the carbon to nitrogen ratio, here's the dry sites, the prairie and ag. Carbon-nitrogen ratio decreases with depth, and that's what other people have found. It doesn't change that much in the dry site. In the wet site, 850 millimeters, what you see here is the native prairie. And in the agriculture site, because we're moving nitrogen down, and I'll show you later, the microbes, they're using the carbon, and so we actually see a lower to carbon-nitrogen ratio in the ag site. Carbon is being mineralized and nitrogen is being conserved to some extent, which is kind of interesting. So again, from a climate change perspective, land use and precipitation is going to greatly affect those dynamics in the soil profile. We don't understand those completely, but it's, I guess, worthy of future research. We looked at enzyme activity through the profile. We looked at two particular key enzymes. Beta-glucosidase is a carbon cycling enzyme, so it relates to carbon availability and mineralization as facilitated by those microorganisms. And then N-acetylglucosamidase is a nitrogen cycling enzyme. If you look at these profiles, they're completely different. This is combine the three land uses. But what you see here is that the carbon cycling enzyme glucosidase pretty much has a uniform profile. As you go deeper, it decreases. But look at the nitrogen enzyme, amidase. The dry site tends to follow the profile of the carbon and the organic matter, carbon and nitrogen that I showed earlier. But at the wetter sites, this is Kanza, 850, this is 1,000 millimeters, there's increased nitrogen cycling enzymes, amidase, deeper in the profile. Again, it's reflecting that more nitrogen deeper in that profile as a result of agriculture and that environment. And so we really need to understand the biochemical and the biology that's going down deep related to precipitation and, of course, land use. And in fact, this is not from this study, but one of my other students, he looked at the profiles of the ratio of carbon cycling to nitrogen cycling enzyme. And what he saw here was the dark circles is cropland, conventional tillage, and what he sees here is that as you go down through the profile, in the cropland it's carbon limited, and as the grass, it's nitrogen limited. Again, if you look back at the grass here is nitrogen limited. So what you were seeing is that deep in the profile we lost that carbon, we conserved that nitrogen, and so it's becoming carbon limited compared to the native grassland system. So now we're switching our nutrient cycling, biogeochemical cycling, as a result of land use, and it'll be interesting to see, and I suspect that the precipitation or climate change will affect that biochemistry down, not only at the surface, but even importantly down to a meter or 2 meters in depth. Nope. One more. Okay. Back one. There you go. Okay, so in the remaining minutes, what I want to talk about is then how can we design systems, manage those soils for better resilience to that changing climate. And so my example is the prairie, the tallgrass prairie. This is the tallgrass prairie at Manhattan, Kansas. Kansas is not flat. People think it's flat, but this is the native prairie. There are certain characteristics about this ecosystem that I use as my model ecosystem. One, it's biodiverse, has lots of diversity in plant cover. It's climate resilient. We have naturally extreme droughts and wet years. That ecosystem has evolved to withstand those weather cycles. It's very water efficient. Those deep roots has different plant species, so it's extremely efficient with the water. And it's also nutrient efficient. It's not normally fertilized, but it sustains itself. And we've done work on nitrogen cycling, phosphorus, and it's extremely efficient with the nutrients. Very conservative, very little losses, but yet it's very productive. So I'm a soil scientist, so the secret is it really in the soil. So this is how I put my research program together to look at soils, soil health. Soil health is a big topic in the United States and increasingly around the world. There are 3 components for soil health. One is the carbon, organic matter content of the soils. The other is some measure of soil structure. We do aggregate stability, but some measure of soil structure. And then, of course, being a soil microbiologist, I think the microbial community in activity is important. But these 3 are interrelated. And I showed you the relationship between mycorrhizal and aggregation. And I'll show you some other data here in a minute. But if we have those 3 components, if we have good soil carbon, good soil structure, good microbial activity, then we're going to have a resilient systems. It's going to be productive. It's going to be provide ecosystem services, and it's going to have resilience, both for the water, but for the rooting depth and other aspects that we want out of that soil. So again, going back to aggregate structure, we're looking at these large macro aggregates greater than 2,000 microns in size. This diagram from Julie Jastrow, it's been used around the world, shows this aggregate structure. And so what we have is these small microaggregates less than 250 microns in size. And these are bound together by fungal hyphae, that's these black strands here, by root debris, polysaccharides from bacteria and fungi. But it's this binding of the hyphae and the gluing together that's extremely important. Inside those aggregates is organic matter, carbon. And so if we build these aggregates, then we're going to trap carbon inside those aggregates. And one of the reasons we get a carbon increase in soils is because we do less tillage, we build carbon inside those aggregates. It's touchy. I got it. Maybe. There. Yeah. Okay. So this is some work that one of my students did, Gail Wilson, back in 2009. It's a classic experiment. What we did was we took different grassland systems and we suppressed the mycorrhizal fungi. And so we have here different levels of hyphae, mycorrhizal or extra radical hyphae out in the soil, and aggregates greater than 250 microns in size. These are those macro aggregates. And what we saw was there's a relationship, a strong relationship between the amount of aggregates and the amount of hyphae in soil. Okay, and just by suppressing the hyphae, we didn't do tillage, we just suppressed the hyphae, and we lost aggregate structure. So it shows the importance of the hyphae in binding those aggregates together. And what we also show is then there was a relationship between the amount of carbon in soil and the hyphae or aggregates. So that's that three-way interaction between the microbes, the soil structure, and carbon. Really tightly controlled. And with climate change, if we lose that connection, we're going to degrade our soils. And this is, sorry. This is illustrated here. And if you take those soils that have less aggregates and less of that binding, you wet them up, what happens is they dissolve. The soils that have that good aggregate structure maintain those aggregates. And so from an intense rainstorm event, those soils that are less fungal hyphae and less aggregates, they're going to wash downstream when we lose our soil. Okay, so what I'm going to show you here is pictures of a no-till system in Brazil. But what we're looking through is the residue as you go through that residue layer. And what you see here on the top of that soil is these white strands. And that's not roots. That's fungal hyphae. Again, the importance of that linkage between hyphae and soil structure. And what we've shown is that less disturbance, less tillage, increases that fungi in the soil, and it improves soil health. So we had an experiment in Manhattan, Kansas. We call it the Kanza, again, the prairie. And we had 3 treatments. Sorry, the translator couldn't translate some of these slides. But we have tilled sorghum, right? No-till, Sambe directo, right? And then the prairie. So here's the replanted prairie. Here's sorghum with tillage and no-till. So we started this experiment in 2006, and we've been continuing that. They're sampling this week. My students are. They should be. And what we've looked at is the change in the soil quality as a result of that replanting of the prairie and the sorghum with tillage and no-till. What we've seen here is that look at macro aggregates in the top 5 centimeters. The replanted prairie has increased in macro aggregates over that 11 years. And this is the native prairie. So we're almost back in aggregate structure in 11 or 12 years to the native prairie with that replanted grass. The no-till increased, but it's basically flattened off, but it's at 50% versus the native is 70%. So we're improving the aggregate structure
in the no-till systems. And the till basically hasn't changed. It started out at 35% and is at 38 or 39%. So we really haven't improved that aggregate in that tilled system. So the goal in some of our other work has shown that no-till is improving soil health and is getting somewhere between close to the prairie. If we look at the microbial community using phospholipids, again, where the bars are the same color, they're statistically same. And if you look across, here's fungi, gram-negative bacteria, gram-positive bacteria, and actinomycetes. The prairie, replanted prairie, and the no-till are more similar. So the fungi decreased, as we saw in the other examples. With tillage, we lose the fungi. The no-till is more similar to the prairie. The bacteria, gram-negative bacteria, the big change is in one of the markers, less of this particular phospholipid in the prairie and the no-till. In the gram-positive, again, the no-till and the prairie are more similar, tilled encouraging more of these the gram-positive bacteria, and actinomycetes are again similar between the no-till and the prairie. So one of the management strategies that we're looking at is no-till because it has less disturbance and acts more like the prairie. So anyway, that's some of the research data. Last year, the National Academy of Sciences Board on Agriculture and Natural Resources came out with a report talking about the science breakthroughs needed for food and agriculture systems by 2030. They have 5 research themes. The one that's relevant to my topic is the opportunity for the soil microbiome. And what they suggested was there was need understanding the relevance of the microbiome for agriculture to harness this knowledge to improve crop production, transform feed efficiency, increase resilience to stress and pest diseases. And so you can download this report, it's in English, download this report, but it lists 5 themes for agriculture, and the microbiome, soils and microbiome is one of those major themes. Soil, sorry, I couldn't translate this into Spanish, too many words, but the sub-themes for the soils area was we need to maintain the soil fertility and restore degraded soils through best practices such as reduced tillage, cover crops, but we need to use new sensing technologies, biological strategies, the microbiome, and integrated systems. Secondly, to increase nutrient use efficiency. Third was to create a more productive and sustainable production systems by identifying and harnessing the soil microbiome. It's a new frontier. And use that microbiome to increase water efficiency, nutrient efficiency, and reduce environmental stress. And the last part is extremely important, is to take those technologies and transfer those to the farmers, to the land managers. That's extremely important. So in summary, am I good on time? Okay. So climate change will likely impact our soils and the soil biology. I've tried to show that. There's some experimental evidence for that. But then if we look at like land use and precipitation gradients, we can see that precipitation is going to be an important driver, and particularly that intensity of that rain precipitation events. And that biology is going to be affected by that climate change and precipitation. We've seen a strong relationship between organic matter, carbon, aggregation, and the microbial community composition, particularly the fungi. I did a lot of my work on bacteria, but I'm realizing the importance of fungi in those systems and important how management can affect the fungal groups. in the land use and the precipitation gradient, we're seeing a decoupling of the carbon and nitrogen cycle in that profile. So while we're looking at the surface, we need to be looking down deep, because that's going to affect environmental concerns and the sustainability of that soil, particularly in permanent or perennial systems. And the enzyme, the biology, we're still doing the phospholipids through the profile, but the enzymatic, the biochemical properties have changed in that profile, reflecting that change in the carbon and nitrogen throughout that profile. And then finally, hopefully it's at the bottom, but soil management is going to be key to rebuilding soil health and providing resilience to climate change. We need to be, at least in cropland systems, we need to be thinking about less tillage, the climate smart agriculture that Miriam talked about yesterday, less tillage, cover crops, rotation systems are absolutely critical if we're going to maintain our soils currently, but in the future for climate change. And with that, thank you. I can take questions, but need translation.
Muchas gracias, doctor Reitz, profesor distinguido de la Universidad del Estado de Kansas, Estados Unidos. Vamos a hacer una ronda de preguntas. ¿Tiene alguna pregunta para el doctor Reitz?
So the practices, as I mentioned, less tillage, so no-till or reduced tillage, that's first. What we've shown is other practices, we can improve carbon, but if you till the soil, you lose over half of your benefit, okay? And then secondly, increase rotation or increase plant cover. So in annual cropping systems, putting in cover crops is extremely beneficial. Also rotations that increase diversity of roots, and more roots is important. So monoculture, crop rotations is better, diverse crop rotations is better than a monoculture. Monoculture soy is terrible. Corn is better, but still it's better to have a rotation in the system. But cover crops, extremely important. And some farmers around the world, the U.S., are actually putting perennials back in the rotation. So they're putting in alfalfa or grass in the rotation, like a 7-year, maybe 3 or 4 years of annual crops, and then 2 or 3 years of a grass perennial, and then they graze livestock on those. And from a nutrient efficiency, I think we need to start looking at the microbiome and the potential benefits of the rhizosphere and the microbes in that rhizosphere. Looking at either adding microbes or mycorrhizae, nitrogen-fixing bacteria to that rhizosphere. Is that you need a translator? Okay, sí. ¿Otra pregunta? Ah, vale.
La pregunta es si ellos, en los estudios que han hecho, tienen un estimado de cuánto tiempo se pueden recuperar o volver a
time.
pueden recuperar o volver a formar agregados del suelo mayores a 250 milímetros.
Okay, you have an estimate of how much it will take to recuperate the aggregates in
Okay.
the soil?
Right. So the question is how much time it takes to reaggregate the soil. So our work shows, well, the one experiment was 12 years. Oh, well, 12 years of replanted the grass. No-till was partway in between. We have an experiment that's 30 years old with no-till, and we're adding compost, and the aggregates are close to the native prairie. So, but that's adding compost, extra carbon to the system. So I would guess 30 to, well, 25 to 40 years, maybe. It depends on the soil. But the short term, you know, even 10 years, we can greatly improve the aggregate structure.
De la precipitación especialmente porque nuestras condiciones en general es muy alta. Entonces, el manejo eficiente de eso que es una variable muy importante de nuestro clima.
¿Lo puedes volver a repetir con pausitas, por favor?
El manejo.
Okay, she is interested in finding out about the management of precipitation. And
Condition of precipitation, how we can manage this kind of relation.
With precipitation.
The tropics, for example.
Oh,
5,000.
yeah, see. So if I understand the question is, how do we manage the precipitation, or the carbon with the precipitation? Is that in tropical systems? Oh, high precipitation, okay. So I work a lot in Brazil, in southern Brazil, Rio Grande do Sul. And our work down there has shown that because of the high precipitation and high temperatures, or warmer temperatures, that we need to, it's extremely important that you do less tillage, no-till. You have the opportunity with warmer temperatures to do cover crops easier than in North America or in the US. And so cover crops, rotation, intensity is important for cropping systems. For grasslands, it's a little bit easier, different, but for cropping, annual cropping systems, you have to, well, yeah, you have to do less tillage, no-till, you have to do cover crops, and increase the rotation intensity. So intensity. So what we do in Rio Grande do Sul is we do wheat, double crop, soybeans, 2 crops in 1 year, followed by a cover crop, followed by an annual crop, maize or soy, and then a third, and then cover crop, and then a third year of another grain crop, soy or corn, maize, sorry. But cover crops all the time, and as soon as you harvest, you plant. So there's something growing on that soil, putting carbon into that soil year-round. That's the advantage, and you would have the same in Colombia, is the temperature and I think the moisture, that you can grow a plant year-round, and that's really key. You want to feed that microbe, you want to feed the soil. Does that understand?
what is your opinion about the process of hydrogen? About the Donald Trump way of thinking.
So what about Donald Trump?
About what he says about
Climate
climate
change. Donald Trump is not a scientist, okay? He can't even read a book. So, it's really interesting in the U.S. because the U.S. administration doesn't believe in climate change or is against it or whatever. I guess I'm against climate change as well, but what's happened in the U.S. is, despite the federal government, the good thing is that cities, states, and companies are getting involved. I work with several corporations, environmental groups, Nature Conservancy, but PepsiCo, Bayer, all those companies are making commitments to reduce their carbon footprint, and they're investing in agriculture to reduce its carbon footprint. So despite the federal government, in spite of Trump, companies and environmental groups, states have put more effort. So maybe that's a good thing, I don't know.
Sí, no, pero lo interesante, creo que todos lo entendieron. Una cosa es la administración de los Estados Unidos actualmente y otra cosa es lo que realmente piensan los científicos y las mismas compañías de los Estados Unidos, que es lo importante. En realidad hay una buena... las mismas compañías saben que tienen que luchar contra el cambio climático y están apoyando realmente la parte de la ciencia. Pero pues ya como todos saben, pues el señor tiene otro pensamiento, pero afortunadamente los científicos y la gente que sí está pensando el clima están trabajando con ese proceso. Good.
All right. Okay. Yeah. So the question is, I focused on the macro aggregates, and basically there's a relationship between the more macro aggregates you have, the less micro aggregates you have. And in our tilled systems, disturbed systems, we see that shift of distribution from macros to more micros. So because we're interested in soil resilience, we want to encourage those macro aggregates to form because they allow for better water infiltration, they protect carbon, and you have less erosion. So we're focusing on those macro aggregates. So it's really a trade-off, macro aggregates or micro aggregates. So now I will tell you that some soils don't aggregate very well. So sandy soils aren't going to aggregate. Silty soils probably will not. Our work in Brazil actually shows, because of the iron and aluminum cementing, that in the U.S., on mollisols, we use 2,000 microns of size as or greater than 250 to 2,000 as our macro aggregate size class. In Brazil, and this probably would be true for parts of Colombia in the oxasols, we actually move up to 4 and 6,000 microns, or 6 millimeters, 4 millimeters in size, because of the cementing. And so we've moved to different size classes in our macro aggregates for our oxasols, or at least my experience. Good morning. So the question was the role of biodiversity in aggregation. That's a good question. So there's several reasons you want biodiversity. A more diverse population is going to be more diverse community is going to be more resilient. But our focus is showing that the fungi, the mycorrhizal fungi and the fungi are really, really critical to aggregation. Partly because of the hyphae binding the soil particles together, but the bacteria also exude glues, polysaccharides that glue that aggregates together. But the fungi seem to be a key indicator, so we focus more on that fungal relationship. But from a soil health or resilience standpoint, you want to have more greater diversity. How much diverse? I don't know. You know, there's a lot of redundancy in that community, but definitely you want a balanced community, whatever that is.
Okay.
Yes, thanks, Sergio. I have a question about the triangle of soil health that you showed. I like very much like the three aspects of soil health. So is the soil organic matter, the structure, and the microbiology. But I was wondering, is there a special reason why you only have the microbiology there? Because is it because you think that the microbes, what changes in the microbes reflects what it changes in the whole community, or in the soils where the fauna do not play such an important role? Because in my experience with soil aggregation, the fauna is also
Right.
the fungi are very important, but the fauna is also very
Yeah. So, so the question was related to my triangle. I call it the Holy Trinity of soils. It's a kind of a joke. But Miriam was asking about the soil biology, microbiology, and how come I didn't mention the soil fauna and those other organisms. I guess I'm a soil microbiologist, so I'm biased, but in some respects, maybe that microbial community composition resents the soil health of the whole biology, but in some of our work, at least in ag systems, that we have reduced soil fauna in our annual cropping systems. So the fungi in particular, but the microbial community is more responsive and more dominant than the soil fauna. I have a colleague that works on earthworms and mites and that they're important in prairie systems. But what I guess our focus of research is the microbiology, and we show that microbial composition. So I mean, certainly the soil fauna are important because they also help aggregate soils, the polysaccharides, and other things. The fungi are critical. They are a key indicator. Okay.
Me interesa como para el público entender la pregunta fue muy interesante y también es dos cosas interesantes ha dicho el profesor Chorrais. Una en el tema de la agregación del suelo y comentaba claramente en suelos arenosos y en suelos que tienen mucho limo es más complicado lograr una rápida agregación, no es tan fácil. O sea, ya en suelos con más arcillas es más fácil lograr una rápida agregación. Ese es un tema que mencionó el doctor Rice. Otro tema interesante también con las preguntas es en el tema de cuáles son esos microorganismos que están trabajando a nivel de la agregación. Crítico la parte de las micorrizas, y lo he mencionado claramente, las micorrizas ayudan mucho en el tema de la agregación y es un indicador que podemos usar también para mirar si nuestros procesos que estamos haciendo en la parte de manejo de suelos están contribuyendo realmente a lograr esa agregación. También menciona que las bacterias también ayudan a la agregación, también son aglutinantes, pero en el tema de la agregación, como indicador, es más fácil, o han logrado tener metodologías para poder mirar como indicador micorrizas. Recordemos que para uno buscar indicadores, pues las variables deben de ser fácilmente medibles y que puedan ser repetibles. Hay variables, hay de pronto elementos claves en la agregación que son importantes, pero que no tenemos metodologías para poder hacer el seguimiento, y entonces eso nos genera mucha variabilidad en la información y no podemos interpretar. Por eso también cuando buscamos este tipo de análisis de variables son que las podamos también medir y que sean repetibles, y ese es un tema muy clave. Entonces, por eso él insiste mucho en el tema de las micorrizas y que realmente participan en el proceso. La pregunta que hace la doctora Mirjam también, bueno, ¿y qué pasó en el triángulo que mencionó? ¿Por qué olvidó la macrofauna? Y realmente todos sabemos que es muy importante en el proceso de agregación. Como mencionaba el doctor Rice, pues ellos están enfocados mucho en el tema agrícola, y cuando uno está trabajando ya en suelos agrícolas, pues realmente el problema el tema es que la macrofauna se ha venido desapareciendo en esos procesos. Cuando trabaja uno ya en pasturas o en praderas, ahí realmente uno ve una muy buena dinámica de la macrofauna porque no ha sido destruida. Y ese es el componente clave también en la parte agrícola, y es cómo vamos a lograr nosotros volver a integrar la macrofauna, porque realmente la resiliencia que estamos buscando no es un solo grupo de organismos, es toda una integración de ellos trabajando en conjunto en el suelo para lograr esa estabilidad. Y esa es la clave, y hago aquí un comentario. Si ustedes han visto, en este congreso le estamos dando muy fuerte al tema biológico. Definitivamente hemos mejorado mucho en la química, en la física estamos trabajando, pero estamos todavía muy pobres en la biología. Y hay mucho por entender, y por eso estamos con estas presentaciones, estos científicos y más, las que ustedes están mirando, es darle más luces para que fuertemente trabajemos esto, porque la resiliencia está en la vida del suelo, que la estamos dejando por lado. Entonces, él reconoce el tema, como le digo, entonces hablaba de la agricultura, más de referente al tema agrícola, porque se encuentra más fácil los hongos en las partes agrícolas y muy poca macrofauna, pero no es que la esté dejando de lado. ¿Alguna otra pregunta? Yo tengo una pregunta al doctor Rice, porque en una conferencia anterior nos había hablado de un indicador que eran los fosfolípidos, y ahora estamos hablando de las enzimas. Voy a preguntar cómo funcionan las dos. Doctor Rice.
The question was, I talked about phospholipid and measuring microbial composition as an indicator of microbial, in that triangle, the microbial component. And then I also talked about enzyme activity. I think, as Miriam was talking about, I think you need to have three measures: carbon, organic matter, soil structure, and we're using aggregation. It seems to really work for us, but there could be other indicators of soil structure. But it's that physical air, gas, or water movement that's really, and rooting, that's important. But the third indicator is the biological. I put microbial, but some measure of biological properties, because I think it's really important to have those three: chemical, physical, and biological. So we're using phospholipids. We've been using it for, 6 years or so. It really relates to soil structure. But it could be some other biological measure, soil fauna, but enzymes is another way to relate to biological activity. And it relates to nutrient cycling, carbon, nitrogen, sulfur, phosphorus activity. So it doesn't, and we're doing more enzyme activity. And so the thing about enzyme activity is you can take soil samples, dry it, ship it, and do the enzyme analysis later. But the phospholipids, you have to freeze the soil immediately and freeze dry it before you ship it. We've gotten samples from Africa, from Brazil, from CIAT, from Colombia, and we've done the phospholipid, but you have to treat it more carefully. The enzymes are more stable. So it's a potential indicator. The Soil Health Institute in the United States is using enzymes, they're using respiration, they may use phospholipids, but I don't know which one is better, but we're using different ones to find out different aspects, and maybe we'll decide on 2 or 3, but it doesn't have to be one. English.